So we're now going to be moving on to a company that we covered recently, which caused quite a stir. These guys are called Senisca.
Okay, so hello and a warm welcome to Cenisco. We're very happy to be talking to you today, and thank you very much for giving us the opportunity to introduce ourselves. So we at Cenisco have pioneered a new approach to treat the diseases of aging based on over 10 years of world-leading academic research. So aging and age-related diseases are driven by the accumulation of senescent cells, at least in part. And removal of senescent cells has been demonstrated to bring about rejuvenation of old animals. Strategies for their removal include senolytic approaches to kill senescent cells or xenomorphic approaches to rejuvenate them. So for some indications, a rejuvenative approach may actually be useful, since many disease-relevant tissues are cell-poor or non-proliferative. And recent data suggests actually that some subsets of senescent cells are beneficial. We at Cenisca are experts in harnessing RNA biology for the rejuvenation of senescent cells. So as we all know, aging is caused by the failure of a few basic health maintenance mechanisms called the hallmarks of aging. And at Cenisca, we have discovered a new hallmark of aging, dysregulated RNA splicing. And we have discovered that by restoring this to more youthful levels, we can rejuvenate cells and target the causes of aging, not just the symptoms. So most genes are capable of producing more than one RNA message according to the needs of the cell. The ability to do this is absolutely crucial for cellular health and for successful aging. And the decision as to which form of RNA are produced from a gene in response to any given challenge is made by a group of proteins called splicing factors. At Cenisco, we have discovered that levels of splicing factors decline with age, but that when we restore them to youthful levels, we can bring about cellular rejuvenation. And this remarkable discovery is the result of over 10 years of world-leading research from the University of Exeter in one of the UK's leading RNA research facilities. So Senisca uses oligonucleotide interventions targeted towards the master control genes that regulate splicing factor levels, or to individual splicing events, reset splicing factor levels, restore splicing patterns, and rejuvenate aged cells. And this is a platform technology that could be applied to many diseases driven by senescent cells. At Cenisco, we will apply these first to diseases with a proven link to senescence, occurring in accessible target tissues, thus amenable to local delivery, diseases with high unmet clinical need. So we've identified the underlying biology and the pathways behind age-related splicing dysregulation, which has allowed us to identify and validate 4 genes, which, when we target them, can rejuvenate senescent cells. We also have further data from which we've identified over 20 other targets for future evaluation. We can design novel oligonucleotide biologics to manipulate the expression of these targets for rejuvenation of cells. We can intervene either upstream in the pathway, which will allow us to affect many targets simultaneously, or downstream, which will allow us very precise specificity to minimize off-target effects. We appreciate that the delivery of oligonucleotides historically has been challenging. However, there have been a variety of new developments in this space over the past years which have removed many of these barriers. So new oligonucleotide backbones and new delivery methods have revolutionized these approaches and made them tractable. In fact, there are now over 10 oligonucleotide biologics which have achieved regulatory approval and are in use in the clinic. Actually, 5 of these have been approved in the past year alone. There are several potential delivery methods available to us, and members of our scientific advisory board have specific expertise in this area. So, like the previous talk, we have a platform technology here, and our initial we could apply it to any disease driven by senescence, but our initial indication will be idiopathic pulmonary fibrosis, known as IPF. Now, 3 million people worldwide have IPF, and there is currently no effective treatment. In addition, drugs that Cenisco will design for IPF will have potential applicability to other fibrotic lung diseases and indeed to chronic obstructive pulmonary disease, which has a much bigger market. Currently, available treatments for IPF cannot stop or reverse disease, but only very modestly slow it, and they don't work for all patients. Cenisco's technology promises to not only slow disease, but even to stop or reverse it with greater efficacy, lower toxicity, and more specificity. So our timeline encompasses two complementary approaches. Our main modality is oligonucleotide biologics. And we have 4 existing targets ready for exploitation and the creation of new IP. As I said, we have 20 further targets which will undergo validation early in 2021. And following target validation, we will take a traditional hit to lead to CAN track through ex vivo 3D cultures, co-cultures of more than one cell type, and tissue models, followed, of course, by evaluation in vivo. Our second modality is a small molecule approach applied to skin aging, whereby we will take an intelligent computational docking-based screen for molecules affecting our novel pathway. And this is a self-contained and targeted piece of work, which has actually been driven by interest from a large US-based corporate potential partner. Our background IP is currently in PCT phase until regional protection starts in January 2021. Future IP will be tailored towards each medical indication and is supported by a strong and established research and development infrastructure, including protected 7-year access to our £30 million plus world-class lab facilities, which are equipped with state-of-the-art, fully maintained capital equipment. This lab access, together with our long-standing expertise in RNA and senescent cell biology and cutting-edge tissue culture techniques, presents an extremely high barrier to entry for potential competitors. A detailed 3-year forecast profit and loss account is available for review. Net operating losses in year 1 to 3 will be funded by seed investment rounds plus non-diluting grants. For our skin aesthetics program, a joint development and global license agreement is already under negotiation with a world-leading US-based cosmetic corporate. Traction is extremely strong in this program, and we also now have a second global player from the anti-aging skincare market entering competitive negotiations for a similar first-look licensing co-development deal. We have low occupancy and equipment costs due to our 7 years facilities agreement and a highly experienced team controlling operating and project costs. So our team consists of myself as CSO, Dr. Ben Lee as CTO, and Kirsty Semple as COO. And between us, we bring a wide range of skills in the senescence, RNA biology, and commercial space. In addition, we have people with relevant and appropriate skill sets on our scientific advisory board, including Professor Steve Wilton, who designed and guided the very first oligonucleotide therapy for Duchenne muscular dystrophy into the clinic, and Professor Merlin Thomas, who is currently working on oligonucleotide delivery to the lung in the context of COVID. So we've recently incorporated, we're brand new, and we're working towards the opening of our launch seed round, which is happening imminently, targeting £950,000 in seed funding, covering a 9+ month cash runway. We welcome discussion on key investment terms and timetables, so please contact one of the team if you are interested. We envisage several exit scenarios. So we could sell or license each indication separately, for example, each pharmaceutical program and our skin aesthetics. And we'll engage in early partnering talks with established pharmaceutical companies targeting cost-neutral research and future royalty revenues. We'll also keep firmly in sight at the right time, of course, either a full trade sale or an IPO as we move forward towards clinical phase for our pharmaceutical program. So thank you for listening. And if you're interested in talking to us further, please contact one of the Cenisca team or indeed talk to us in the breakout room.
Awesome. Well, thanks very much, Lorna. So we have a little bit of time to field some questions. So I've got a first question for you in relation to your R&D pipeline and going beyond that initial phase. Where would you get off? Would you hand over to a big pharma or another organization or some form of third party to help you with your development timeline, or would you see yourself going all the way through when you go through future funding rounds?
Sure. So we're not going to limit ourselves at the moment, because I think a lot of it's going to depend on what we find and how it goes. Certainly for our skin aging program, that's a defined piece of work that we will be working in a co-partnership deal. We envisage that we will do the initial work. They will then take the reins and take that through the trials. In terms of the pharmaceuticals, again, it depends on the situation we find ourselves in, and we're leaving our options open.
Good. And in terms of the skin products, is that something that you'd see yourself going out of pharma and maybe into some of the big cosmetics companies as partners?
Well, I think the way we see that going is that once we've got it to a certain point, we will hand it over to them. We will obviously, they will have a license to use it. We will get royalty revenues from that. But I think where our hearts lie are in the pharmaceutical arena. Of course, skin is the largest organ in the body. It's accessible, and it's a really good initial proof of concept, proof of principle for us for our approaches.
Excellent. Very good. Well, we have no further questions coming through on the chat channel. So how's the funding going? Are you making progress?
We're getting there, yes. So we're early days. We're in negotiations, and as I said, I think it's a question of watch this space. We're hopeful we'll fill our funding round, and we're just really looking forward to getting going.
Very good. Great. Okay. Well, wonderful. So, Lorna, thanks so much for your time today. Great presentation.
No problem. Thank you for yours.
I'm sure that presentation, and I'm sure there'll be some people wanting to speak with you in the breakout room. So that's great. Well, thanks very much.